And so we want to resolve these fragments that we made. Hopefully we only made what we PCR'd, right? And what we amplified, that fragment size that we're looking for. But it's also possible that our primer sat elsewhere and that we need to resolve what we are looking for away from anything that should not have PCR'd but might. So that's all it's telling us right here, is that we're going to separate DNA, RNA, or protein according to size. So you can do any of these by gel electrophoresis. We're going to be just talking about DNA today. And so we need a bunch of stuff. And so first, right, we'll just talk about this, the gel loading solution. If you think about, we need to have our sample be a little bit heavy. So we use something that has glycerol in it. And then we need the agarose, we need a buffer, we need some way to be able to make it happen. We need some way to generate that electrophoresis. And then pipettes, and then some way to see DNA, right? We're not gonna be able to see it with the naked eye. We have to stain it to make sure it's actually visible. So if we talk about just a little bit of basics about DNA electrophoresis. So how does this work? Well, like I said, we want to add what's in our PCR tube, hopefully is our PCR product, and we want to put them into little wells or like holes, a depression in our agarose gel, which is essentially like Jello, but it doesn't have sugar and dye in it. And then add an electrical current to make it pass through this matrix of gelatin. Okay, so here's our setup. So here's our little gel guy, and here's our wells, like little depressions, so we can put liquid in there. But this gel is also sitting in that buffer that allows the electricity to pass through the gel. And so if we don't add something to our little tube of PCR to make our liquid heavier, thicker, denser than the buffer, it'll just float away if you try to just squirt it down into a hole. It just comes out. So we add that glycerol to make it a little heavier, and we add dye, some stain, some color, so that we can tell which wells we've actually pipetted something in. A clear liquid into, you know, a clear gel, you're not going to tell you might be adding stuff all over. And so once we've loaded it, we add a current. We have the negative diode over here and the cathode, the positive charge on this side, and that will pull negatively charged molecules down out of the wells. And since DNA is negatively charged, it will run to the positive cathode, because opposites attract when we're talking about negative and positive charge in chemistry. And so at this point, we can't see any of the bands. That's why they're showing them all grayed out. What we would be able to see is the dye that we added. So they should have had a little picture. There's a dye that's orange, and it runs it runs through the gel, usually smaller than your DNA. There's also a gel that's kind of purpley, and that usually runs with smaller fragments of DNA. And then there's a teal colored, my personal favorite, which usually runs when you're running a gel up here. So in general, in my lab, we put all three in. And so you have a teal guy, a purple guy, and an orange guy. And you try not to run the orange off the gel. That way you know where, how long you ran your gel and where approximately your bands should be. So you can use those dyes to help you decide where you would expect to find your 500 base pair piece, depending on the equivalent base pairs that dye runs to. But usually we run a molecular weight ladder, and there's a whole bunch of different ones depending on what sizes you're looking for. But in general, you could get one that has a piece that's 10,000 base pairs, and then maybe a 5,000, and then a 2,000, then a 1,000, and then 500, and then maybe 300, something like that. And so these would essentially what that lane looks like, would run through your gel, and that's your standard. Then you can compare where your bands are to determine what size they are. So for example, I have no idea what, they just made this up, but like, let's say that one is 500 base pair molecular weight marker that we know is 500 base pairs, and the one above it, let's say that one is 800, then what would we guess that that guy has to be, if that's the unknown, the experimental? Well, it has to be between 500 and 800, right? Does that make sense? So because it's not great resolution, all we can really say it's between 500 and 800. If it's exactly halfway between, then maybe we'd say it's 650-ish, right? But if we really wanted to know, we'd have to resolve with a different kind of gel and have a marker that has more than just an 800 and a 500 piece. But that's just the general idea. And so we've stained the DNA in order to see it, so we can actually see those bands on a gel and see the molecular weight marker, so that you can actually do this. So there's a bunch of different types of stains, which we'll talk about, I think, today, if I'm remembering correctly, and which ones we use and why. So that's just the overview. And we'll get into a little more of the nitty-gritty as we go. Does anybody have any questions? I think it's pretty basic. We're going to talk more details, so if it gets a little crazy, don't panic. No time to panic. Ah, an ant. Oh, giving away the farm here. Come on. Okay. All right, my little friends. So this would be what you need to do to prep for gel electrophoresis. Now, you can also purchase the pre-made gels, precast gels, but they're not that cheap, and then you have no choice of what percentage you're going to make your agarose. So it's so easy to make agarose gels that precast is really, really, really for super lazy, or when you need something to be super consistent. You don't want any human error. So if you're doing something for product development or anything like that, then you'd want to have precast, because then they're guaranteed to be exactly the same or so forth. Otherwise, I feel like it's just a waste of money. But that's me, I'm cheap. Okay, so let me stop here really quick. I'm gonna stop my recording and restart. I'm gonna pause my recording. All right, now we're recording again. Okay, so let's go back to what our equation was. So we have to make the buffer before we can, right here, add the agarose. We have to make this liquid. Right? Nobody gave that to us. So, but what we were handed was a 50x concentrated solution, and we have some water. And we're gonna make 100 mils, and we want it to be at 1x at the end. So what does that tell us about what we want? Remember, we always have an equation, and we can make it what we want and what we have, right? M1V1 equals M2V2 instead of start and finish. Like, what do we want? What do we need to have? So what are the givens? We always want to have 4 things given. No, we always want to have 4 items, 3 of which are given, and we only have 1 variable. Otherwise, we can't really do this equation, right? So what do we have? We have total volume. We know it needs to be 1x, and we know we start with 50x. So that's 3 of the 4. Okay, so if we say we know we want 100 mils at the end, right? That's what we want to end up with. And what concentration do we want that at? 1x.
1x.
Yeah, 1x. But what do we have? Do we have it at 1x? No, we have freaking 50x. So then our question mark, or what we need to find out, is how much of that, right? Like, and our units are in milliliters. So it's got to be something milliliters. Question mark milliliters. And so then we just do the stupid math, which again, if we want to be anal about doing the math, we do the same thing to both sides. So we divide both sides by 50x to leave our question mark all by himself on the other side of that equation. The 50s cancel. 50 divided by 50 is 1. X divided by x is nothing, right? They go away. And then 100 times 1 is 100 divided by 50. What's 100 divided by 50? Ah! Isn't that 2? And what do we have left? We got rid of this guy, our units, our mils, right? And so then our question mark equals 2 mils. And then we just have to make sure, well, if that goes over, there we go. Our 2 mils is the question mark that went with our 50x, right? That tells us we need 2 mils of 50x buffer. Again, we want our total volume to be 100 mils. And our other issue is water. So then how much water do we need?
98 milliliters.
Give me a thumbs up if that is okay. No? That's not okay? Oh, I see a thumbs up. Oh, yeah, I see thumbs. Okay, thanks. Sorry. I know sometimes it's hard to find your thumb. So that would be 100 mils, I guess I should finish that, of 1x buffer. Yay! Okay. So that's getting to our buffer. Now we need to make our agarose. So we're never going to make agarose in water because we want it to be in the same buffering solution, the same concentration of buffer as what we're going to put in the running tray, which we haven't talked about yet, but trust me. In order to get that current to flow through, right, we have to have buffering capacity. And I'm sure you guys did buffers in chemistry, and I tried not to ever remember how that worked. I did the equation so I get the answers right, but like whatever. So we're just taking chemistry's word for it that we need a buffer in order to send current and not use up all of that ability to pass that current through the gel. So the gel has to have a 1x concentration, and the buffer you run it in has to be 1x. So if one is 10x and one is 1x, it's screwed. If you messed up the math on one or the other and you have a half x and a 1x, it gets all messed up too. So we want the gel that we're making, and the agarose is going to be the solid stuff, right? And then we want the liquid it's sitting in to pass the current through that at 1x. You can make it at 2x as long as both are at 2x. You could do it at half x. If both are half x, it just wouldn't be a gel you could run for hours and hours and hours. It would use up the buffer, the molecules in there allowing that to happen. So 1x is what they figured out is what you need. But as long as it's the same, you could run it in 2x, you could run it in 3x. Probably more than that, it probably gets screwed up. But the gel and the buffer has to be exactly the same. See, there's that lawn guy. Now he's on the other side. It's like the gardeners are after me. One neighbor and then the other. Okay, so now we need to know how much agarose we're going to put in here. So for right now, we're gonna go with, you shove the agarose in there, the right amount, and you swirl it up, and it's not gonna dissolve. It's not going to look like when you put salt in water and you first see the crystals, and after you swirl, 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 they go away, and it's a completely, you know, it's in solution. It will look like a slurry. It'll look like you can see the little chunks of agarose in that liquid. So just like Jell-O, if you've never made Jell-O, you're gonna have to go buy some Jell-O and make it, just so you get the idea, and eat it, because Jell-O is freaking good. You have to boil it. In order to get the agar in agarose or the agar in Jell-O to go into solution and then be able to go from that liquid to a solid, you need to boil it. And so what we do is we just boil it in a microwave, and we're just super careful, and we heat it little by little, because you want to take it out and swirl and then add a little more and swirl, because you can get something called superheating, where if you don't swirl the flask in between, somewhere in the words it tells you like every 30 seconds or 20 seconds or whatever. You just kind of watch it. You have to swirl it carefully, because with that, the slurry in there, it sort of suppresses boiling. And then when you pick it up and swirl, it causes the boiling to happen, and it can spew out all over and burn the living crap out of you. So anytime you're making agarose, you always hold that flask slightly away from you and not at somebody else. I mean, we're 6 feet apart now in labs, but before, somebody might be right next to you. You can't hold it to the side and swirl it, because you're gonna burn the hell out of them. And I know this because I've burned the hell out of myself before. Because no matter how careful you are, at some point, you're gonna grab the stupid thing and swirl it without paying attention. So it superheats and it spews hot molten lava all over you. Okay, so you swirl, 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 and then you're supposed to let it cool to about 60 degrees. Some people actually stick it in a hot water bath that's at 60, so that it can't drop below 60, especially if they're not ready to pour a gel, or if they're just being super anal. I always did the rule of thumb, whereas if you could pick up the agarose flask barehanded, once you're used to hot stuff and you've like burned off a bunch of stuff by frostbite on a -80, then it's ready to pour. The problem with pouring too early is that it can actually melt the tray. I mean, they're super hard plastic, but it can totally melt your the thing to make wells. So you don't want to pour it like boiling right out of the microwave. So you let it cool for a bit. You keep swirling it to make sure it's, you know, an even solution. It's not chunkier on the bottom than on the top. And then you put together your tray. So the tray is usually a little piece of clear plastic. Sometimes they have these little dams that you stick on the end. Old school, you put a piece of masking tape over it. And then they have a little notch where you can put the wells in. If you forget to put that little guy in there, you're kind of screwed. You got to start over. Because if it cools without it and hardens and it has no holes, then there's nowhere to stick the DNA in. So this leaves those little slots where the DNA goes. And so then once it's all set up, then you pour your gel into that guy. And then it says, right, here we go, we're pouring, and then we wait 20 minutes. It depends on the size of the gel. If it's a mini gel and it's poured pretty thin, it's about 20 minutes. If it's a bigger gel, it takes longer. If you're in a super big hurry, like when you're a PhD student and you're trying to freaking graduate and get your publications or whatever, you can actually pour the gel in a refrigerator. So as long as the fridge has a nice tray that's, you know, like a solid bottom rather than, you know, if it, you don't want it to fall through, that's what I'm saying. Then you can actually pour it in the refrigerator and it cools down much faster. So that can get you going, especially like normally we would do this while our PCR is running. We have 2 hours in a PCR machine. You get your gel ready, boom, you have plenty of time. It's totally cool, it's great. If you forgot and you're like, oh crap, you don't want to spend an hour waiting because you've got to get to class or whatever you're doing, then you can do it in the fridge. Just a little fun fact. Okay. So here's words, just tells us stuff. Yes, yes, yes. Okay, questions? We're good? That makes sense? Like, it's hard to, unless you've actually done it, but, you know, we can probably find some video somewhere of somebody actually doing this so that we can look at it. I'll try to find that later. Or one of you guys can for me. Okay, so now we're gonna go back, I think. Whoa, cheating. Hang on. Let's see what's next. I can't remember. No. Where did I put it? No. 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 Yes. Okay. So we're going to talk here. So we're going to get into how much agarose do we need into that flask. Okay. Remember, I just said put whatever you're supposed to put in there. So how do you know what you're supposed to put in there? So that's where knowing our fragment size and possible mispriming will really help you. So most people, if nobody tells them anything and you're running a diagnostic gel and the person told you make me an agarose gel and then they left, you would make a 1%. That's right in the middle, pretty safe. You know, you don't have to wait till they come back. They're ready to, they want their gel and you didn't make it because you didn't know what percent. So that's the safety percent. Now, I'm sure this is online somewhere, but this is like the ancient chart that I had taped to my bench way back when. I took a picture of it. And the type of gel doesn't really matter. This was just the company that we used, and it's just your standard molecular biology grade agarose. So just regular old agarose. This guy, this Metaphor agarose, is a specialty agarose just to look at little pieces. So in my graduate work, I had to clone lots of small fragments, so 150, 50, 200. Those are not easy to resolve on a regular agarose gel. And even if you can't extract them out of a really high percentage gel. So there's a specialty agarose that can resolve fragments, you know, really, really super small, which was super nice. But we don't need to worry about it, but that's just why it's on this chart. And the other thing people, before this type of agarose existed, you had to use acrylamide, which can resolve down to like several base pairs, but it's really hard to extract DNA out of, and you have to use all kind of harsh, nasty chemicals that, you know, like are on the carcinogen list and all that stuff, and hazardous waste and blah, blah, blah. Whereas from agarose, all it is the DNA dye that's a problem. So anywho, then we got to think about what buffer are we using. We said 50x running buffer to 1x, but there's actually two main types of buffer. One is called TAE, one is called TBE, and TBE is the one you use for your standard diagnostic gel. It has the best buffering capacity. So that's the one you would use just to look and say, yes or no, I have my PCR product. If you want to extract DNA or RNA out of gels, then you would use this TAE. Tris acetate EDTA versus Tris borate EDTA, I couldn't even remember. It's still a decent buffer. It just doesn't have the buffering capacity of TBE. And it's a little more of a pain in the ass to work with, because the acetate part will become volatilized in the microwave. So you lose some of the actual acetate comes out, right? So just for standard gels, we use TBE. If we're gonna extract something, then we go to TAE. And it matters because the resolving power is a little bit different. So what this chart shows us is what percent resolves what types of fragments of DNA, what sizes, I guess I should say, not types, what sizes. And so if remember I said 1, if you're doing that 1% agarose, that guy resolves best between 1,000 and 7,000 base pairs. So that's generally what people are PCRing, right? We said we wanted our fragment to be, you know, at least 500, but it can be, you know, up to 1,000, 1,200, so forth. So that's your standard. But like for me, mine, if you remember, my fragment was going to be 500, right? Which falls into the 500 to 4,000, or even, right, between 300 and 3,000. So if I ran a 1% gel, my band's gonna be a gooey mess, not gooey, but like a smeary, that's a better word, a smeary mess at the bottom of the gel. It may actually, because it's not gonna resolve well, it may be mixed up with primer dimers, which are always a nice glowing band at the bottom of your gel when you do PCR. We'll talk about that when we get to there. So it would kind of suck, and I might not be able to see anything. So I'd want to run between a 1.25 and a 1.75. And because I know how it works, I would be fine with doing 1.25% agarose. So for yours, it depends on what size your PCR product is. And so that's why I wanted you to have your primers handy with telling you what size your PCR product is, because that will determine what percent gel you would want to make. Oh, and then just down here, this is just an example, and it's because I use metaphor all the time, that's why this one was tied to my bench and not the regular, but it's just telling you, remember I talked about the two different dyes, one was purple and one was turquoise. Like you can look up for regular agarose where these guys actually resolve to, and remember I said there's also an orange one, which is actually smaller than those guys. And so if we were running this metaphor gel and we were running it in TBE, this turquoise guy would run it like at 310 base pairs, and the purple would run at 40 base pairs. And so that's just a nice way while you're running the gel to know that if you're looking for a band of 200 base pairs, it's going to be between these. And so you'd want sort of like if this is your gel and here's your lanes, right, and we're going this way, we might want, right, we want the turquoise dye to run right here, and the purple dye to run right here, sort of trisect the gel, and then we'd know, we can't see it while we're running it. Remember, we can't see DNA until we've dyed it, and we dye it after we run it. So we know ours is right in the middle. And then like, let's say you left the machine on and you didn't remember the timer, and you come back, you're like, holy crap, I got caught up in something, and you run your purple completely off, then you're gonna be like, oh God, what if I ran my, you know, my stupid bands off too? So, you know, so it gives you an idea of where you're going, especially if you're not sure how long to run something. It's like, it's my first gel. Do I run it for 1 hour, 2 hours, 3 hours? Right, so then you can watch it to make sure you don't just put all your DNA into the buffer on the cathode side, right? It's been done. And again, in the good old days, well, they weren't good. The bad old days, the old power supplies didn't have timers. So you had a timer. And you didn't have a phone. Yeah, thank you. And so you couldn't set a timer on a phone, so you carried around a nerd timer when you went to lunch or whatever with a count. And I walked uphill through the snow with that timer. Okay. So does that make sense? You can't just pick some agarose off the shelf and jam it in there. You have to make an educated decision to make sure you see what you need to see. Okay. Okay, my little friends.
Dr. Malone?
Yes.
I put a link in the chat. It's a pretty cool page, and it has a short video with they're running a mini gel, and it's like 2 minutes and 30 seconds long.
Yay! You're awesome. Thank you. Okay, so now let's go back to this little picture so we can watch that little guy right after class, or, you know, in a breakout room when you're bored because you're done. So now we have, what do we have? So now we put our tray, the whole tray with our gel. We took the ends off because we want to make sure the current can run through. We stick him in this, the setup, I don't know what, whatever. We pour in our 1x buffer. So we used buffer to make our agarose, and then we also pour it in here, right? And then while it's in there in the buffer, we load it. So they're not showing us, but you'd be loading it in there while it's underwater, under buffer. Then you add the lid, right? Tells you positive and negative, and the positive side's always red. And so in the lab, and again, at some point, most of these trays are now, or these setups are now foolproof, because the lid doesn't fit backwards. But again, in the bad old days, we didn't even have a lid. We just had an electrode thing where you could accidentally electrocute yourself if you stuck your finger in the buffer and turned it on. And so you'd set it up, and invariably you'd set it up backwards, and then you'd run your DNA off the gel the other direction into the well. So the rule of thumb was always to watch until your DNA dye gets into the gel going in the correct direction. And so we always said, run to red, back to black. And so if you said that in your head and you looked at your gel, then you wouldn't do it backwards. But we'd still watch it to get in to make sure we didn't totally screw it up. And then as you see, here's our little power supply. You plug that bad boy in. You set it for the appropriate voltage or amperage, which you can look up and figure out. It depends on the size of the gel, how many volts or amps you can jam through that guy. And so then you figure that out. You set it up, set your timer, and you're good to go. So let's, I forgot. Go back. Oh, hello. All right, so let's, yeah, go away. Just shove him right off the screen. How about you? No, he's stuck. How about you? How about you? No, okay. All right, so I wanted, what did I want? I wanted for my agarose in my gel, right? So this, what's in here, I wanted 1x TBE. So we're pretending now I can tell you this is 50x TBE. And so we made a 1x. And then I also want it to be a 1.25% agarose. Okay, that's what I want. So that's what I want to, I want to put enough in here to give me 1.2% agarose, and then we go to the microwaving. So we need to remember something I told you, I kind of remember now how many weeks ago, so I'll tell you again because I wouldn't remember. We need to remember when we were talking about weight and volume and concentrations that 1 gram in 100 mils equals 1%. So they're doing that weight per volume in grams. And so we pretty much use that for any, whatever it is, that it's just designated, that's its percentage. So everybody knows 1 gram of whatever it is 1%. So 1 gram of NaCl in 100 mils of water is a 1% salt solution, right? 1 gram of agarose in 100 mils is a 1% agarose solution. So it's really easy if you're making a 1% gel. You put 1 gram in 100 mils. If you're making 100 mils, it's easy. If you're making 1,000 mils, you're making a liter, which that'd be a big ass gel. But what if you're making 200 mils? What if you're making 30 mils? You need to know how to do that little setup, right? So Okay, everyone, so for our example of the setup to figure out percentage, we will use our equation, the 1.25 grams in 100 mils, right? So we know the answer should be. And so we'll use this setup to do your equation as well. So what we're going to see is, as we know, or may or may not know, we have 125 grams in 100 mils. How many grams in 100 mils do we need to get the percent that we want? And so we're going to do our math, whereas remember, if we need to cancel stuff, we have to flip the ratio, right, to make it go away. And so what do we get then? We get our 100, ooh, I didn't carry my units, 100 mils to go away. We're not going to get rid of our x's even though, right? Maybe that's not exactly set up right. Okay, so I digress. We're going to get rid of these, right? And we're essentially just left with, you know, x equals 1.25 grams because everything else canceled. And that's our answer, right? We already knew that 1.25 grams in our 100 mils was going to give us 1.25%. So we just showed using this equation how to do it. So let's try it with 1.25% in 30 mils. So you guys are in your breakout room, you're gonna do this and then come back and I'll help you. And we're back. Well, we're gonna pause because somebody's talking during my video. Okay, nobody's talking during my video now. All right, okay. Friends, we're back. We never went anywhere, but we're back. Okay, so let's do our setup. 1.25 grams, 100 mils. We know that is 1.25%. And then what grams are we going to use to make 30 mils? And so we're going to do just like we did before. Hope it works. Carry my units over x. And do the math. Here we go. These guys are canceling. 1.25 times 30. Let's see. I gotta get my calculator. 1.25 times 30 equals this. 37.5 divided by 100 equals, right, x equals 0.375 grams. That makes sense because it's got to be about a third. If it was 1 gram, it would be a third. So that's about a third. We want 1.25. We got our right answer. Okay, what about for our TBE? We also had that question. So we were making, we want 1x in 30 mils. We have 50x. Our question is, what do we want? And so we take our 50 on each side, and we do our math. And so we have question mark equals 30 divided by 50 equals 0.6 mLs. And that's of 50x TBE. And that means we need 29.4 mLs of H2O. So that's how you do it. You guys are good. Nice job. Thanks. I thought so. recording. So we're going to just talk really quickly about visualizing the DNA. Remember we talked about you can't see it when it's running unless you put the dye in the gel, and I suggest never ever do that, because then everything that gel has ever touched is contaminated, and all the buffer is contaminated, and then you have to have liquid waste, solid waste, everything in your lab you ever touch needs to be in gloves, and so you can't trust anything. So it's better to stain after. I know labs stain before, but I don't recommend it. It also does change how DNA runs through a gel when you put that intercalating dye in, so it does change the molecular weights a little bit, although as long as you dyed your marker, then, you know, or it's in the gel, whatever. So the main point here is that we shouldn't trust pretty much anything that stains DNA, because except for, I think, this blue stain, and then there's something called gel red, everything else intercalates into the double strand. What does that mean? That means it gets in between, like in the major and minor grooves of DNA. And then it fluoresces under UV light. So there's not, so it's an intercalating agent. The thing that allows us to see it is its ability to fluoresce. But it's not the fluorescent part that's the problem, it's the intercalating part. So anything that intercalates into DNA can't be good. It's automatically a mutagen. It automatically causes problems in DNA. If something gets in there, it can mess up your base pairing. It can mess up your actual bases, right? So yank parts off, like you lose a methyl group, you lose a hydrogen, right? So that's why ethidium bromide is bad, and that's why all stains you should use gloves. So ethidium bromide is a standard. We try never to use it if we don't have to, but it's the most sensitive of all the dyes, and it's a known mutagen. They came out with this cyber safe. People got all crazy about it and thought they didn't need to wear gloves, even though you look and it says wear gloves. And if you look at its safety, it just hasn't been tested how bad of a mutagen it is. So they name it cyber safe. It's not safe. It's an intercalating agent that fluoresces. So it might be better than ethidium, but still you need to do all the precautions you should. I don't know anything about this blue, flash blue, and gel red is not supposed to be a mutagen, but you still gotta wear gloves because you just can't really necessarily trust anything. And so what we do, I think, let's see, they show us, oh, whoops, wrong one. So, oh, this is their instant stain. But however you do it, it's after you run it, you take the gel off the tray. You don't want the tray to get soaked in the ethidium or the CyberSafe or whatever, because then that thing's contaminated forever, and you have to be super careful. That way, like, somebody washes, you know, you wash your trays and all that, and you hang them up to dry, and somebody grabs one without a glove, it's bad. If you've never seen ethidium, then it's fine, or one of the other things. So in this case, they're showing us you can just put this little piece of paper on it, have some ethidium soak in to your gel, and then all you have is that little piece of waste and your gel is waste, versus we kind of put them in a big, like I don't know if there's a picture, a tray of, yeah, like this, like they do for the CyberSafe. You make it, it comes concentrated, you make a 1X solution, you put your little gel into the tray, put that stain in it, and let it sit. So we do that with ethidium. I think that's how we do gel red, but I don't even know now. And so that's the standard of how to stain. Yeah, and that just tells you what. And then it's just telling you about their Insta Blue, same idea. Their Flash Blue, which is super expensive. I looked at it, but it doesn't take very long. I get their Insta Blue takes overnight or something, but it's not a hazard. So for classes, right? And then that's it. This is just the end of their little promotion, and just pictures of some gels with different molecular markers and so forth. So Yeah. So I will post a little tutorial about agarose and our calculations so we can watch it again, since like I did it wrong for you and taught you wrong, and that's all bad. But now we're really going to remember better because it was all wrong. And then next time we will look at some real gels, hopefully, and do some analysis. So we will pretend like we ran our PCR, and hopefully our fake PCR will work. And then we'll move on from there about what are we going to do now that we have our PCR product, as far as cloning it into our PCR vector and then moving it into our final vector. So does anybody have any questions, comments? Oh, watch the little video in the chat if you have time right now, or copy and paste that link to watch another time so you can see it. And I'll try to pull some other stuff from real live lab, since we're not in a real live lab. Are we okay? It was a lot. It was all right.
Oh, it's good.
Okay.
Nice refresher.
Nice. All right. Everybody else okay? Yes.
Thank you, Dr. Malone. Have a nice day.
All right. You guys too. Thank you so much. I will see you next time. I'll post some stuff for you.
Thank you,
Okay,
Dr.
thank
Malone.
you.
Thank you. Bye.
Bye, guys. Love ya.
Bye.
Do something fun or eat lunch or something.
Professor Malone, I had a question.
Okay.
It wasn't in regard